Ever find yourself wanting to really truly grasp a complex science topic? You get the deep understanding without drowning in dense textbooks.
Yeah, happens all the time.
Well, that's exactly what we're aiming for today. We're doing a deep dive into something absolutely fundamental in biology, how our cells control which genes get switched on or off.
It's a huge topic. It's basically how you get different cells, like skin versus brain, and how cells react to, well, everything. Crucial stuff.
Exactly. Think of this as your fast track. We've got this source material, Sayasot text, which goes into all the intricate details.
Right, and our mission really is to pull out the core concepts from it, give you a solid foundation in gene regulation.
And the text suggests a key starting point, maybe the key starting point, is controlling when RNA transcription even begins.
That's definitely where a lot of the action is, the initiation of transcription.
Okay, let's get into it then. Transcription regulators.
Hmm.
The text mentions these. They're proteins, right?
Yeah.
What are they actually doing? Are they like the librarians deciding which genetic blueprints get used?
That's a great way to think about it, actually. Yes, they are proteins, and their job is to bind to very specific stretches of DNA.
Specific stretching.
We call those cis-regulatory sequences. And by latching onto those, these regulators are gatekeepers. They decide if a gene gets transcribed into an RNA molecule or not.
And the text points out something pretty striking. About 10% of all our protein coding genes are just for making these regulators.
It's a huge number, isn't it? It
Yeah.
really tells you how vital this regulation is for, well, everything a cell needs to do.
So it's a major part of the genome's job.
Absolutely. And they often don't work alone. Most bind to the DNA as pairs, either two identical units, that's a homodimer, or two different ones, a heterodimer.
Like little teams.
Exactly. And they recognize those specific DNA sequences using particular shapes, structural motifs on the protein surface. Think of it like a key fitting a very specific lock on the DNA.
Molecular keys, I like that.
And it gets even more layered. There's this concept of cooperative binding. Sometimes one regulator binding makes it easier or harder for another one to bind nearby.
Ah, so they influence each other.
They do. And this ties into how DNA is packaged up too, wrapped around proteins in those structures called nucleosomes. That packaging itself adds another layer of control over whether the DNA is even accessible.
Okay, so we've got these protein gatekeepers, these regulators,
Yeah.
binding to DNA, sometimes working together. How do they actually flip the switch? Turn a gene on or off? The text hints it's different in bacteria compared to us.
That's a really important distinction, yeah. In simpler organisms, prokaryotes like bacteria, it's often more direct.
Simpler setup.
Kind of.
Yeah.
The regulators tend to bind very close to where the enzyme RNA polymerase needs to start working, right near the promoter.
Okay.
And depending on who binds where, they can either help polymerase get going, that's activation, or physically get in its way, blocking it, that's repression.
Seems logical. Relatively straightforward.
Mm-hmm.
But eukaryotes, for example, the text says it gets.
Messier. More complex, certainly.
Yeah.
Yeah, eukaryotic gene regulation is significantly more intricate. You can have lots of different regulators controlling just one gene.
Lots of cooks in the kitchen.
Sometimes. And crucially, these regulators can bind to DNA sequences really, really far away from the gene's starting point, the promoter.
How far are we talking?
Tens of thousands, even hundreds of thousands of nucleotide pairs away. Those DNA building blocks.
Whoa. Hundreds of thousands. How can something binding way over there affect what happens way back here at the start of the gene?
That's where the physical nature of DNA comes in. It's flexible. It's not a rigid rod.
Ah, okay.
So the DNA can actually bend and loop around, bringing those distant binding sites and the regulator sitting on them physically close to the promoter and the RNA polymerase machinery.
Like reeling in a long fishing line.
That's a good image for it, yeah. It allows for long-range communication along the DNA molecule.
So these distant regulators in eukaryotes, they aren't usually doing that direct blocking or helping, like in bacteria.
Not typically, no. Their mechanisms are often more indirect. Eukaryotic activators and repressors frequently work by changing the local environment of the gene.
Oh, so?
They can recruit other proteins that modify the chromatin structure, that DNA-protein complex. They might make it more open, more accessible for transcription, or clamp it down, make it inaccessible.
So remodeling the whole area.
Exactly. They also influence the gathering of the core machinery, the general transcription factors, and RNA polymerase itself at the promoter. They do this by attracting helper proteins called co-activators or co-repressors.
It sounds like a whole committee has to agree before anything happens.
It really can be like that. And the key point from the text is that it's the specific combination of all these regulators present in a cell at a given time that dictates the final outcome.
Which specific regulators are active?
Yes, that combination determines when a gene is turned on, where, in which cell type, and how strongly it's transcribed, the rate of transcription.
That makes sense. It allows for nuance.
Absolutely. This combinatorial control is fundamental. It's how we get the staggering variety of different cell types in our bodies from the same basic set of genes.
Right. The text dives into that, how a skin cell is different from a neuron because they're expressing different gene sets. And that difference comes down to which transcription regulators are active in each one.
Precisely. Each cell type has its characteristic set of active regulators, pushing the expression of genes needed for its specific job. A muscle cell needs muscle proteins, a liver cell needs liver enzymes, and different regulators orchestrate that.
It's like tailored instructions for each cell type.
Exactly. And what's also interesting is that one single type of transcription regulator might be involved in controlling many different genes, and it might be active under different circumstances or in different cell types.
So they're versatile, not just one regulator, one gene.
Definitely not a simple one-to-one map. It's much more networked.
Okay, that covers how different cells get their identities. But the text also brings up cell memory. How does a cell, say a liver cell, stay a liver cell after it divides? How does it remember?
That's a critical piece of the puzzle for multicellular life, isn't it? How do tissues maintain themselves? The text highlights a few mechanisms. One really important one is feedback loops.
Feedback loops.
Yeah, particularly positive feedback loops. Imagine a transcription regulator. Let's call it regulator A. Suppose regulator A not only turns on other genes needed for the cell type, but it also turns on its own gene.
So it makes more of itself.
Exactly. Once it's initially switched on, perhaps during development, it ensures its own continued production. So when the cell divides, both daughter cells inherit some regulator A, which keeps the cycle going.
It's self-perpetuating, a molecular memory circuit.
Precisely. And these kinds of transcription circuits can do more than just maintain a state. They can perform other simple logic operations too, leading to more complex decision-making within the cell. And these circuits don't exist in isolation. They're wired together into huge, complex regulatory networks. These networks are what drive incredibly intricate processes, like the entire development of an embryo from one cell.
The complexity is just staggering. Now, besides these regulator feedback loops, the text mentions another layer of memory, epigenetic inheritance. That sounds like inheritance beyond the DNA sequence itself.
That's spot on. Epigenetics refers to heritable changes in gene function that do not involve changes to the underlying DNA sequence. It's about how the genes are marked or packaged.
So, information passed down through cell division, but not in the A's, T's, C's, and G's.
Correct. The text gives some key examples. One is DNA methylation. This is where a small chemical tag, a methyl group, gets added directly onto some DNA bases, usually cytosines.
And that tag matters.
Oh, yes. Methylation patterns can influence gene activity. Often, high methylation in a gene's promoter region is associated with silencing that gene. And importantly, these patterns can be copied when the DNA is replicated during cell division.
So the silent state gets passed on.
That's the idea. Another powerful example is inherited states of chromatin condensation. Think about the inactive X chromosome in female mammals.
Right, one X gets shut down in each cell.
Exactly. That X chromosome gets tightly packed into a structure called heterochromatin, silencing most of its genes. And this highly condensed inactive state is stably passed down to all the descendants of that cell. It's a form of cellular memory.
Incredible. And the text also mentions genomic imprinting.
Yes, imprinting is another fascinating case, primarily in mammals, also often involving DNA methylation. For a small number of genes, whether they are expressed or not depends on which parent you inherited them from.
Mother or father.
Right. The gene copy from the mother might be silenced by methylation while the father's copy is active, or vice versa. This imprint is set during egg and sperm formation and usually maintained throughout life.
So, parent-specific gene usage.
Essentially, yes. All these mechanisms, feedback loops, DNA methylation, chromatin states, imprinting, they contribute to the cell memory, allowing specialized cells to maintain their identity. It's crucial for building and maintaining complex organisms.
Okay, so we spent a lot of time on controlling transcription initiation, the very start, and these memory mechanisms. But the text is clear. Regulation doesn't stop once the RNA is made.
Absolutely not. That's a critical takeaway. Transcription initiation is often the main control point, but there's a whole suite of regulatory opportunities after that. Post-transcriptional control.
What kind of things are we talking about?
Well, there's quite a list. Sometimes transcription can start but then get halted prematurely before the full RNA is made. That's called attenuation.
Cutting it short.
Right. Then there's alternative splicing. Many eukaryotic genes have non-coding bits, introns that need to be cut out, and the coding bits, exons, stitched together.
Splicing, yeah.
But the cell can choose which exons to include. From one gene, you can generate different mRNA versions by splicing differently. This means one gene can code for multiple related proteins.
More variety from the same gene. Clever.
Very efficient. Then there's control over how the end of the RNA molecule is processed, the cleavage and addition of that poly A tail. Even the RNA molecule itself can be chemically modified, edited, changing the information it carries.
Editing the message after it's written?
In some cases, yes. And then there's controlling if and how the mRNA gets out of the nucleus into the cytoplasm where the ribosomes are.
So, export control.
Yep. And even once it's in the cytoplasm, the cell can control where it goes, localizing specific mRNAs to particular spots. Think nerve cell extensions needing certain proteins right there.
Makes sense. Direct delivery.
And then, of course, there's control over translation itself, how efficiently the ribosomes initiate protein synthesis on that mRNA.
Starting the protein-making process.
Exactly. And finally, a really key one, controlling how long the mRNA molecule sticks around. Regulated mRNA degradation.
How quickly the instructions get shredded.
Precisely. A short-lived mRNA will produce less protein than a stable, long-lived one. And the text notes that many of these processes involve specific RNA sequences or structures being recognized by regulatory proteins or even other regulatory RNA molecules.
Which brings us nicely to that last major topic in the text, those non-coding RNAs. We know about tRNA and RNA for protein building, but there's apparently a lot more RNA out there that doesn't code for protein.
That's been a huge area of discovery. We now appreciate that cells make vast numbers of non-coding RNAs with diverse functions.
Okay, like what?
Well, a well-studied example is RNA interference, or RNA. This involves small guide RNAs. You might hear terms like microRNAs, miRNAs, small interfering RNAs, mRNAs, or Peewee interacting RNAs.
Small RNAs doing big jobs.
You could say that. These small RNAs use base pairing, like the ATCG rules, to find specific messenger RNAs.
They target specific messages.
Exactly. And depending on the specific type of RNAi and the match, this pairing can lead to the mRNA being chopped up and destroyed.
Silencing the message.
Or it can block the ribosomes from translating it, or even trigger changes in chromatin to shut down transcription of the gene itself, promoting heterochromatin formation.
So they can interfere at multiple levels.
It can. It's a versatile system. And interestingly, similar systems in bacteria and archaea act like an adaptive immune defense against viruses, using RNA guides to target and destroy foreign genetic material.
Wow, an ancient defense mechanism. And the text also mentions LNCRNAs. Large non-coding RNAs.
Yes, LNCRNAs, or long non-coding RNAs. This is a huge and diverse class. We've only recently started uncovering them, and honestly, for most of them, their functions are still pretty mysterious.
Still figuring them out.
Very much so. But some have been shown to act as scaffolds. They don't code protein, but they can bind to multiple other molecules' proteins, other RNAs, and bring them together in the right place at the right time to carry out a specific task. Like molecular organizers.
Bringing the team together? Okay, wow. Let's try and pull this all together then. It's clear that controlling gene expression isn't simple. It's this incredibly complex, multi-layered process.
Definitely not just flipping a switch.
Right. Starting transcription is a huge control point, with those transcription regulators being key for making cells different and letting them respond to things.
Absolutely. Combinatorial control is central.
And then you have these amazing memory systems like feedback loops and those epigenetic marks, methylation, chromatin changes that let cells remember who they are through division.
Crucial for development and stability.
But it doesn't stop there. Regulation continues after transcription with splicing choices, mRNA stability, translation control.
A whole cascade of potential adjustments.
In this whole fascinating world of non-coding RNAs like the RNAi system and these mysterious LNCRNAs, adding yet another layer of control.
It really highlights the interconnectedness, doesn't it? How all these different levels work together to fine-tune everything so precisely. It's not one master control, but this intricate, dynamic network.
You know, digging into this, it just makes you appreciate the sheer elegance and complexity humming away inside every single one of our cells. We just sort of exist, but underneath, There's this unbelievable molecular orchestration happening constantly.
It is truly awe-inspiring when you get down to the details. The precision is remarkable.
It really is. And it leaves you thinking, doesn't it? What happens when these super precise regulatory systems go wrong? How does that lead to disease or problems in development?
That's exactly where a lot of research focuses, understanding those disruptions.
And thinking about those non-coding RNAs, especially the LNC RNAs we know so little about,
Hmm.
what secrets do they still hold? What new biology, maybe even new therapies, could emerge as we unravel their functions? It feels like there's still so much to discover about how life manages its own instruction manual.